Hello, this is Dr. Griffin, Assistant Professor of Biology at Tuskegee University. I will be presenting to you Module 1, Identifying and Studying Life of Biology 230. Module 1 focuses on the major concepts of Chapter 1 in your Campbell Biology textbook. You need to read all of Chapter 1, but should focus your studying on the concepts discussed and tested in Module 1. Notice that many terms and phrases are in bold in this presentation. These are terms, many which are in your book, that will aid you in maximizing your mastery of skills and concepts associated with this module. You should use dictionaries and other references to define words that are not familiar to you, bold or not. Well, with that, let's start Biology 230, Cell and Genetic Biology. Notice that the meaning of biology is the study of life. Well, what does study mean? Well, as many professors think of it, study represents a passion for something. In fact, study comes from the Latin word to have zeal or passion for. So I just want to remind you that throughout all the contents of this lecture and course is to encourage you to have zeal and passion for this subject, or at least have zeal and passion for your life. In order to study life, we need to find a working definition of what life is. We need to know what it is that we're about to study. Take a minute to see if you can come up with a concise definition of life. Pretty hard, huh? Currently, there is no universally accepted definition of life. However, scientists recognize that there are properties of living things that can be determined to give us a guideline to determine the living from the non-living. These are considered the classic emergent properties of life. And when I say emergent, I mean that there's more than one part, that these properties arise from the interactions of more than one part. Even these emergent properties are found in single-celled organisms, such as bacteria. Let's quickly talk about these emergent properties of life. The first you see on the left is order. All living things are thought to be highly ordered or categorized. In fact, there is some unique complex organization to each type of life, each organism. Living things also exhibit the trait of evolutionary adaptation. That means that they have either created or they have been passed down traits or features that allow living things to have a best fit to their environment. The next emergent property of life also deals with the environment, the responding to the environment. In fact, your nervous system is responsible for allowing you to sense and to respond to the environment. A living thing also is the product of reproduction. And after reproduction, living things will go on to grow and develop. Another emergent property of life. All living things are categorized or thought to process energy or metabolize in some way, such as this hummingbird in the picture, who has a high metabolic rate. And last but certainly not least, all living things are thought to have some way to regulate, or this idea of homeostasis that we'll talk more about in Biology 120. as seen in the hare here that uses its long ears to regulate body temperature in hot, dry environments. Another distinguishing feature of living organisms is the presence of cells. A cell is the smallest unit of life. You can think of a cell as the Lego of life. Cells must have at least two things, one being a cell membrane and the other being some type of nucleic acid, specifically deoxyribose nucleic acid, or DNA, as I'm sure you know of it. Now, according to the cell theory, all living things must contain cells. 2, that cells come from pre-existing cells. 3, that all the vital functions of an organism or a living being occur within the cells. So that means that everything that keeps you alive actually happens within these small cells. And last but not least, cells contain hereditary information, allowing genes to be passed down from generation to generation. Now, there are two major categories of life, and there's actually three major domains that we'll talk about in this chapter. But these two major categories are defined about the type of cells that these organisms possess. And those would be prokaryotic cells and eukaryotic cells. Prokaryotic cells do not have membrane-bound organelles. This includes the nucleus. Prokaryotes will have a cell wall. They will have a nucleoid, which is just a region that contains the DNA. And prokaryotes will have ribosomes because they need to make proteins. Eukaryotes, on the other hand, will have multiple membrane-bound organelles. And in fact, plant cells, which are eukaryotes, will also have all the membrane-bound organelles and cell walls. Now, that we've taken time to think about defining or differentiating living things from non-living things, let's have a quick thought exercise. In fact, this thought exercise will be very helpful in preparing for exam 1. Here on this slide, we have a picture of a fire as well as a picture of dormant spores. Well, fire is ordered in some degree. It responds to the environment. It metabolizes, it processes energy. It can be regulated in some way. There's a good chance or a good argument you can make that fire is alive. Now, most of us would not agree with that. But would you consider fire alive? It has many of the emergent properties of life. Or look at this dormant spore on the right. It's not moving. It's not growing. It is not responding to the environment. In fact, only once activated will it grow and develop and be able to reproduce. But we all know that spores give rise to what we consider life, the plants, the ferns that we see. I highly recommend that you go to the websites listed on this presentation slide to discuss more about how to define or differentiate life from the unliving or the non-living. Well, besides defining life, scientists also struggle to classify life. And what scientists and biologists in particular have come up with is a certain taxonomy. And taxonomy only means the order of things. And we use life classifications just to make it easier for us to study life. Notice I've broken up on the left the major groups in which we can classify life, starting with domains, which we have 3 domains, which the eukaryotes and versus the bacteria versus the archaea. Those are the three domains. And after the three domains, which contains all of life, we go down a little smaller to the level of kingdom, and then to the level of phylum, class, order, family, genus, species. And in fact, as we go down this list of categories, notice that the organisms are becoming more and more alike. Similar ancestors is the main component of ordering these species or these organisms into these different groups. In fact, life is grouped by behavioral similarities, morphological similarities, as well as molecular analysis. And this is the key, especially in this day and age, because genome sequencing is being used to determine if two organisms are part of the same species or not, or same genus or family or order, class, etc. And so we actually are using DNA or DNA similarities, or even maternal DNA or other types of RNA species to look at how organisms have evolved over time and be categorized into these different classifications. A good phrase to remember these levels of organization or classification is Domineering King Philip came over for good steak. That will give you all the major classifications of life, from the domain, so the highest level of classification, to species, which is very specific, or the lowest level of classification of life. As I continue with this first module lecture, I want to take time to focus on the fact that science is a process. It is an act. It's something you do. It's not merely showing up to class and being present. But if we're going to do science, if we are going to be engaged as young scientists, which you all are, know it or not, we must be ready to come to perform. We must come ready to do. Science comes from the Latin scientia, which means knowledge. So we have already started on our process or our career of knowledge seekers. Thus, the major goal, my major goal for you during this course, is to give you the equipment, you, the young scientists, to be mastered up with the knowledge and the tools to discover new knowledge, either very applied knowledge, specific knowledge, or very pure basic research knowledge. And you can use that with your peers and discover that with your peers. I actually encourage you all to work together, both formally and informally, to help each other through this class and all your courses that you may struggle with. You, the young scientists, must start asking questions. And part of asking questions is the first step of the scientific method. The scientific method, as we will come to know in the next slide, is critical to minimize human bias. Here is a flowchart of the scientific method, which is a hypothesis-based science. It is a hypothesis-based discovery tool. Notice that it all starts with a question. That's the first step in the scientific method. You must be curious. I want you to ask questions, not make requests. Let me give you an example of what I'm talking about. Here's a request that I've heard many times. Can you tell me the answer? Request. Now here's a question. How is ATP formed from sugar? Notice the difference. Questions seek knowledge. Requests fulfill a temporary desire. I'm here to help you for the long term, and to allow you all to become lifelong learners. So, let's start asking questions. Let's stop making requests. Okay? Back to the scientific method. So here we ask a question. Next, you're going to do your background research. So you're not going to ask a question and just go right into the lab. You're going to ask a question, then do background research, and then you're going to construct a hypothesis. Next, you're going to test with an experiment, then analyze the results of that experiment, draw conclusions from those results, and those conclusions and results will either support your hypothesis or they will negate your hypothesis. In both occurrences, you should report those results formally and informally. Now, if your hypothesis is proven to be false or only partially true, then you should think. Try again. Do more background research. I know the arrow is going to construct the hypothesis level, but I encourage you to go more background research and then make a new hypothesis and continue with the scientific method. Like I said, the scientific method is a hypothesis-based science, which Chapter 1 does talk about. It, in fact, is an example of deductive reasoning. Deductive reasoning is using a general principle to make a specific prediction or a set of specific predictions. Again, I'm going to reiterate. Deductive is going from general to specific. General to specific. A well-constructed hypothesis that will be used for hypothesis-based science in the scientific method leads to predictions. Therefore, you can do an experiment. And this hypothesis and its following predictions are testable. And what we are testing is if the hypothesis is false or true. Now, after you have tested your hypotheses, plural of hypothesis, if your hypothesis is supported and confirmed by others, not just your lab group, but by all others in the nation or the world that studies something similar, then we can derive what we call a theory. And theories are formed after a hypothesis has been tested and supported / accepted by many groups. Research is really what the word says. In fact, let's take a minute to recognize the RE or the re in research. And this is so true. Remember from your days of prefixes that re means to do again, repeat. And so literally, research means to search again and again and again. The interaction between a hypothesis and a theory illustrates this point very well. Once a hypothesis is supported by experimentation, a theory can be created. Once a theory has been validated and accepted by many, it will be used to explain phenomena. A good theory will lead to many more hypotheses, and thus many more theories. Let's look at the figure below. You see, we have an initial hypothesis. After doing experiments and repeating these experiments or replication of these experiments, our hypothesis say it's supported. Once supported, it will lead to a theory. Once the theory has been accepted, it will spark new questions. And these new questions, remember scientific method, will lead to novel, new hypotheses. And thus we have this whole circle of research continuing again, from hypothesis to theory, to new questions, to new hypotheses, and it would continue over and over again. Okay? Now let me give you a clear example of deductive reasoning. In fact, if the phrase if ... then comes into your mind, it's probably an example of deductive reasoning. Pictured on top here on the slide is an image from a mouse brain that has been treated and infected with a specific type of engineered rabies virus. Rabies virus will go into the neurons and either glow green or red, and it actually is tracing two different neural pathways, and you can see that quite nicely here. Green is one pathway and the red is another pathway. So if neurotropic, meaning viruses that like to infect neurons, if neurotropic viruses infect neurons, which are specialized cells of the nervous system, this is premise 1, and the rabies virus infects neurons, premise 2, then the rabies virus is a neurotropic virus. We have gone from if blank, then blank. This is an example of deductive reasoning. Another way to conduct science, besides the scientific method and hypothesis-based science and deductive reasoning, is discovery science. And discovery science really is what the name tells it. You're going out just trying to discover. It relies heavily on inductive reasoning. Discovery science is based on descriptions of natural phenomena. One of the classic examples of a discovery scientist is Jane Goodall, featured in your chapter 1 of your textbook. She was a famous primatologist who made many specific observations about chimpanzees. That was her expertise. In fact, Jane Goodall was a former professor at large at my alma mater, Cornell University. And you can see here Jane Goodall is taking the pictures of the chimpanzees here, the primates here, and uses in her notebook the pictures and descriptions of the interactions that she noticed and observed. Dr. Goodall used inductive reasoning. She used repeated, specific observations to form general conclusions. Okay, that's the difference between inductive reasoning and deductive reasoning. Inductive reasoning goes from specific to general. Okay? Both types of reasoning, that is inductive and deductive reasoning, can use qualitative and quantitative data. These are the main forms of data or results that can occur from scientific approaches. Qualitative data paints a picture. Qualitative data is full of descriptions, not measurements. So imagine if you're Jane Goodall, you notice that a young chimpanzee is stroking the head of his father. You've painted the picture. But you can even make a better picture by adding more details of the environment and the way the young monkey, the young chimpanzee, strokes the head of his father. Quantitative data uses recorded measurements, often presented in tables and graphs. So I, Jane Goodall, or you, Jane Goodall, could combine your qualitative assessment of your young chimpanzee stroking the head of the father, but someone else is recording the number of times or the frequency or the velocity of those strokes. And therefore we've also entered now quantitative data into the assessment. Again, qualitative and quantitative data can be used both for inductive and deductive reasoning. The power of quantitative data is that it allows us to use statistics to see if our results are due to chance or if we have a real biological phenomenon occurring. After critiquing and analyzing data, scientists will then form models to understand phenomena. My hope is that you, students of Biology 230, begin making models to help you learn in all your science classes, especially this one. Models are representations of natural phenomena. They can be diagrams, 3D objects, mathematical equations, computer programs, etc. In fact, I'm sure there will be types of models that are just unimaginable at this day and age that you will become masters of as you grow as future scientists and doctors and lawyers and psychologists and animal scientists, veterinarians, etc. In biology, models are very useful in teaching and learning how components work together to regulate life. I'll give you a quick example of a model that I studied a lot while I worked to obtain my PhD. I studied female sexual behavior, and one classic stance of receptive females was this behavior called lordosis. And it's actually regulated by a set of neuronal cells that are part of this pathway that you can see here in the picture. And this is just a view of a rat brain, a cartoon of a rat brain. Notice that there's 3 discrete areas that have been implicated in the regulation of the lordosis pathway. Now, obviously there's multiple cells involved and there's different connections that are here that aren't represented in this model. Models are focused and fairly reduced, but what they allow us to do is to teach and learn and focus on the part of the pieces that we're interested in. And with that, I will be ending my lecture on Module 1, covering Chapter 1 in the Campbell Biology textbook for Biology 230. After this model, you should be very comfortable in doing the following. And I underline the word you because I really do mean you. Not your friend, not your book, not your notes, not your teacher, not your TA, but you need to be able to do the following things listed on this slide. And I want to read them to you because I want you to know that they're important. If you can do these things, you will do very well on the tests and in this class. But again, there are things that you should be able to do without assistance in the end. So that means you need to get all the assistance now so that when it comes exam time, you and your brain, if they're not the same thing, are doing them. Number 1, recognize why it is hard to generate a definition of life. 2, utilize the emergent properties to classify living material from non-living material. Use the cell theory to differentiate living and non-living material. Identify a cell. Distinguish a prokaryotic cell from a eukaryotic cell. Understand the basis of how life is classified. Know the importance and role of DNA. Generate more active and participatory roles in your science classes. Employ the scientific method. Compare, contrast, and differentiate deductive reasoning and inductive reasoning. Discuss strengths and weaknesses of discovery science and hypothesis-based science. Understand the powers and limitations of qualitative and quantitative data. Identify the need and basis of creating biological models. It is my hope that you will use not only this lecture, but the videos, the at-home virtual labs, all the assignments and quizzes and tests, and even additional quizzes and tests and resources that you find online or in person or what have you, to be able to comfortably master all these objectives. Now, if you have any questions, or you are finding out that you can't master these objectives very well in a given time frame, and I'm estimating about a week or so for one set of objections for each module for this course, feel free to email me. My email is ggriffin@mytu.tuskegee.edu. It's here highlighted in the presentation. In fact, if you click on it, it's hyperlinked. It should directly open up your Gmail or your email account if it's open, and my email should pop up right there and you can send it to me. Also, I'm encouraging you to post questions on the discussion board on the Blackboard website. Stop by my office during my office hours, or email me to make an appointment so that we can actually get together and I can help you master these objectives. Have a great day, and I look forward to seeing you in class.